Children, now I am operating Nischals lens 3D model on interactive flat panel. You are observing a beautiful 3D diagram of light microscope, and this light microscope is also called as compound microscope. And there you see prints of eyepiece and head is there, and objective lenses are there, and you see the prints of a stage, and you are observing the prints of illuminated reflector and also base arm. So these are the parts of compound microscope. So already we discussed what are the parts of a compound microscope. Again we are observing using, that is, Nischals 3D application, and here you are clearly observing. So I am rotating this compound microscope, and this rotation also helpful to, so this to see the different parts of compound microscope, and it can magnify the object size up to 4000 times using light. So here this is using light, and this light is useful to focus on the specimen, thereby we can see the specimen under objective lenses. So this is compound microscope. The other name for compound microscope is light microscope. Now I am focusing another microscope, that is electron microscope. Children, we discuss that there are two types of microscopes are given in our textbook. So that is compound microscope. The other name for compound microscope is light microscope, and it can magnify the object size up to 4000 times. So here you are observing another microscope, but this microscope is not having the parts of light microscope or compound microscope. Here you are observing electron microscope, and this electron microscope is using electrons by the presence of what electromagnets, and these electrons are useful for to see the object size up to 1 lakh time. So this is electron microscope. A beam of electrons we are using to see the object, to make the object size up to 1 lakh time, and this is electron microscope. This is electron microscope, and it is useful to see the smallest parts which are present in a cell. Now we are observing another one, that is prokaryotic cell. So already we discussed organisms cells are of two types, and they are prokaryotic cell and also eukaryotic cell. So it is a bacterial cell, and children, here you are observing bacterial cell, and it is having flagellum. It is having flagellum, and you see the presence of cell wall. So here bacteria is having cell wall, and it is giving strength, it provides rigidity, and below the cell wall cell membrane is present. And here why it is called as a prokaryotic means it is not having a developed nucleus, but genetic material is present. And here you can see the presence of a chromosome, and that is called as nucleoid, and it is spherical structure, and we can see the presence of cytoplasm. So this is a prokaryotic cell, example bacterial cell. Children, so in this app we observed there are three types of 3D models First one, light microscope. The other name for light microscope is compound microscope, and we also observed electron microscope, and they are using electron beams to magnify the object up to 1 lakh time. Light microscope useful to see the magnify the object up to 4,000 times. And also we observed that is a prokaryote cell on the interactive flat panel. Here you are observing the presence of two scientists, Schlieder and Swan. So they are famous scientists. They proposed cell theory. So how the cells are formed. So they proposed cell theory. According to the cell theory in the year 1838 to 1839, they proposed that new cells are formed from pre-existing cells. So here, when there is the presence of pre-existing cells only, new cells are formed, and these pre-existing cells, they undergo mitotic cell division, undergo division, and finally they give rise another other new cells. So that was the theory proposed by Schlieder and Swan. So here Schlieder and Theodor Swan proposed the cell theory for the first time. The cell theory can be stated as follows. So here I told you living organisms consisting of cells, and cells are called as structural and functional units of living organism, and they are performing different life activities. So here the cell is the basic structural unit of living organism. Next, and here new cells are formed from the pre-existing cells, and that is proposed by Schlieder and Swan. And an organism has its body composed of cells. A single cell transferred life from one generation to the another generation. So these are the that is the information, and this information was provided by Schlieder and Swan in their theory, and they propose that cell is the structural functional units of living organism, and new cells are formed from the pre-existing cells. So in this way, organisms are formed and organisms are developed. So this was the theory proposed by Schlieder and Swan in the year 1838 and also 1839. Thank you, children.